Well, it's a pleasure to be here today, and thanks very much for the invitation to come and speak. I have to do two things. I have to teach a course on genomics and do it quickly. So you're going to be getting Genome 101, and then I'm going to discuss progress and the concepts and the ideas behind the Scleroderma Association of BC research project. Okay, I'd like to acknowledge my co-researchers. Of course, you've heard Dr. Dunn's name, physician. He's the academic project lead. We also have people from the society involved in the research project, and I'd like to acknowledge Mr. Robert Buzza, who is the user project lead, Dr. Raywin Brody, Dr. Robert Holt, Dr. Chris Ryerson, a name you've heard already, Dr. Pierce Wilcox, I think some of you also know Pierce, and Ms. Roseanne Queen, who is the representative for the user partner organization, which is the Scleroderma Association of BC. Okay, on to the course, Genome 101. First question, raise your hands. Is scleroderma an inherited disease? Hands up if you believe it is. There are a few. Okay. No, it's not an inherited disease. That's a surprise to some, I know. How do we know this? Twin study. A twin study? This was done and published in 2003 by Dr. Carol Falagi-Bostwick. Carol in her study found in only one set among 24 monozygotic, so-called identical twin pairs, that at least one twin with scleroderma did both have scleroderma. In only one set among 18 dizygotic, or so-called fraternal twins, with at least one twin with scleroderma, did both have scleroderma. So let's review this. The concordance rate for the monozygotic twins is 1 out of 24, or 0.042. 1 out of 18 dizygotic twins both had scleroderma, and that works out to be 0.056 is the ratio. So for there to be evidence that scleroderma is an inherited trait, there would need to be greater concordance in the monozygotic or so-called identical twins, and we don't see that. So from this study published in 2003, scleroderma is not an inherited disease. It's more likely to develop after an environmental trigger. But are some individuals more genetically predisposed than others is an interesting question. Okay, here's another question. Is scleroderma a genetic disease? Hands up if you believe that scleroderma is a genetic disease. Just a few again. Yes. It's not an inherited disease, but it's a genetic disease. Why is scleroderma a genetic disease? All diseases are genetic, is the short answer. Did you know that? How so? Well, from our parents, we receive DNA, deoxyribonucleic acid, arranged in 23 pairs of chromosomes. Okay, here's another question for the studio audience. What is a gene? Well, it depends on what you mean. It could be a location on a chromosome, in which case it's called a locus, or it could be a specific sequence of DNA, in which case it's called an allele. So genetics is the science of loci, the plural of locus, and alleles. So we talk about genetics. There's two things going on, and you need to get it clarified. Is someone talking about a location on a chromosome or the DNA sequence in a specific allele? That makes life a little bit hard and a little bit confusing outside and inside the discipline of genetics. So here I have a figure. I don't have too many. On the right, I have alleles, and they're color-coded to indicate that they're different segments of DNA, and they're just sitting out there. And then on the chromosomes, okay, we've got loci where you find specific alleles. So that's the picture I want to leave you with about how we connect alleles and loci with the chromosome. Location on the chromosome is called a locus. The actual chemical composition is called an allele. Inherited disease is caused by extra alleles, possibly even an entirely additional copy of a chromosome. It's also caused by missing alleles or possibly an entire chromosome. Inherited disease can also be caused by displaced alleles. They're complete and full, but they're located elsewhere on the chromosome or perhaps on a completely different chromosome. And when either of those conditions occur, we can see disease being caused. Inherited diseases are caused by alleles that were altered in an ancestor. Alleles present but turned off can also cause genetic disorders. And so-called junk DNA, which we don't think does anything, can become switched on and also cause a genetic disorder. So on that slide, you've got the entire complexity of inherited disease. There's a lot of ways things can go wrong. What types of altercations occur to alleles in our own bodies or have occurred in our ancestors? Well, there could be missing segments. There could be replicated segments. There could be altered segments. There could even be changes in allele compression. Allele shapes can be different in men and women. And alleles can have different effects depending on whether it was inherited from your mother or your father. How are alleles altered? Okay, I think you'll be able to answer this question. Radiation. Another. Food. What's in food? Chemicals. So radiation, chemicals. Anything else? Stress could cause release of certain chemicals in our body, which then alter genes, yes. Environmental. Well, chemicals are in the environment, radiation's in the environment. Cigarette smoke, there's another chemical there. Obesity is causing different changes inside the body in terms of chemical composition again, and that affects things. Yeah. There's one that's missing. Medications are chemicals for the most part. This one's kind of a tough one at the back. Mutation? Yeah. Well, these things are causing mutation, so an altered allele is a mutated allele. But there's... That covers a broad spectrum there. It's like environment and bad luck. Yeah, that's pretty inclusive. I think there's also some overlap there, too. Want a Venn diagram for that? That's my mathematics background. No one said virus. Don't we have vaccines now to prevent cancer? And they attack and target specific viruses and build up our immune systems? Yeah. So that was the tough one. I know that. I figured everyone would get radiation and chemicals. We tend to understate and underestimate the effect of viruses. So that's an important environmental trigger, and it's your bad luck if you get one. Well, it's your bad, you'll probably get one. The question is, the bad luck comes in where your immune system doesn't clean it out of your system or put blocks on it very, very quickly. Here's the other point. DNA doesn't replicate all by itself. The steps of the double helix staircase of DNA are broken by DNA polymerase. RNA polymerase makes a copy of the DNA allele in the form of messenger ribonucleic acid, which we call mRNA. Transfer RNA, or tRNA, captures the corresponding amino acid from the ribosome in a cell in order to make the copy. And microRNAs, also called miRNAs, regulate messenger mRNAs by cutting them into pieces when they're not needed anymore. So a microRNA is basically an off switch. Okay, so this slide has a depiction of the process of DNA replication. Okay, over here on the right, we've got DNA. This pink stuff over here is to represent DNA polymerase. So that cuts the double helix. And once the cutting's finished, you're left with a single string that we call RNA. And this is what gets duplicated. You just can't take this and duplicate it like that because it's all tightly bound up together. So we copy DNA by copying RNA. And it's a complicated process, and the brakes are put on the process by microRNA. And we do inherit and pass along microRNA. It's a very stable form of RNA. You're looking at a very short string of molecules put together, and it's very, very stable. So, messenger RNA, or mRNA, and microRNA are popular targets in cancer immunology. miRNA wasn't discovered until 1993. The first human disease found to be associated with microRNA dysregulation was chronic lymphocytic leukemia, CML. A known mutation in MIR96 causes hereditary progressive hearing loss. Miragen Therapeutics Incorporated of Boulder, Colorado, has initiated a phase 1 clinical trial of a compound they call MRG-201. Okay, it's coded, so we don't really know what it is. But we know what it does, or what it's supposed to be. It's supposed to be a synthetic mimic of miRNA 29B, which was discovered at the University of Zurich in Switzerland. And it's intended to treat skin fibrosis in scleroderma. So a phase 1 study is a first-in-human study. Okay? And in this study, controls are being assessed for safety. So in other words, individuals without scleroderma and skin fibrosis are receiving injections, or will once they've been consented into the trial, to see what happens. We're not going to try something in somebody who has a disease first, generally speaking. We want to see if it's safe before we try it in somebody with a disease. So if it gets through that part of the phase 1, a small number of patients with scleroderma will receive injections into normal skin as control, and injections into fibrotic skin over a period of time, and they will be observed to see if there's a beneficial effect due to this compound MRG-201. There is a clinical trials identifier for this, so if you go to the clinicaltrials.gov database in the United States on the internet and plug in that clinical trials number, you'll be able to pull this study up. And the interesting thing is this study is being done in Montreal. It's not being done in the United States. It's not being done in Europe. It's being done by Inoderm Research in Montreal, Quebec. So we're at the forefront of research in this country in scleroderma in using microRNA to treat skin fibrosis. Question I have in my mind, and the question that Tiffany might have, is can it also be used to treat fibrosis of the lung, or do we have to use another compound? And how would we structure a study so that that could be done? Now, I want to talk about the 2-stage GWAS in systemic sclerosis. That's the name of the study, or actually research program. It contributed samples and scientists to the 2 articles that I've just mentioned. Principal investigator is a name some of you might recognize. It's Dr. Maureen Mays, a rheumatologist at the University of Texas, Houston. National Institutes of Health funding for this project from 2008 to 2014, nearly $2 million. Additional NIH funding, $4.4 million, in support of this was directed to the Center for Research Translation in Scleroderma, again located at the University of Texas, Houston, and that was for the period 2008 to 2010. An additional $1.4 million approximately was also received from the NIH for the study known as the Gene Expression as Predictors of Outcome in Scleroderma, which is known by the acronym GENESOS. Total NIH funding in US dollars, $7,770,000. This is a huge study. with a large amount of funding. And I've just looked at the source from the United States federal government being the National Institutes of Health. I went to the public register. I haven't checked to see how much this study or these papers received in terms of funding from other agencies, and the Scleroderma Foundation in the United States comes to mind. Unfortunately, so far, no new therapies to date from genome-wide association studies. So I've got a question for you. Why? Well, an allele codes for a protein, but proteins from other alleles interact, so it's very difficult to predict a biochemical effect of a single protein. Alleles for cystic fibrosis were sequenced and published from the University of Toronto in 1987, but still no new therapies from this. We're learning a lot about the disease, but we're not able to produce a cure. Here's another reason why we're not seeing cures from genome-wide association studies. Only an association between a trait and a loci is found with allelic SNPs. The actual allele involved in the biochemical pathway may not be the loci encoded on the microarray chip, but just in the neighborhood. The closer loci are together, the more likely they are to be inherited from the same parent. And we know that scleroderma is not an inherited disease. So that concludes Genome 101.
Are autoimmune diseases familial? Like our family is riddled with autoimmune diseases, but they're all different.
That's something that we classically see, that you can have a scleroderma case in a family and not see another one, but you might see rheumatoid arthritis. You might see ankylosing spondylitis. You might see psoriatic arthritis. So we do know that autoimmune diseases can cluster in families, but they're not highly specific for a specific disease. And we can also see relationships with other immune disorders, for example, like asthma. Okay, there'll be time for more questions, but I think I've got to move on to the second topic, and that's to discuss the Scleroderma Association of BC research project. So this is a collaboration of scientists at the University of British Columbia and Northern British Columbia, St. Paul's and Vancouver General Hospitals, the BC Cancer Agency, and the Michael Smith Genome Sciences Center. And of course, it's being done in collaboration with the Scleroderma Association of BC. So what are we doing? We're examining the expression of microRNAs in fibroblasts, which are the building blocks of fibrosis in skin and other tissues. And you've already heard earlier this morning from Dr. Winstone that one of those other tissues is the lung. Mononuclear peripheral blood cells, and something called exosomes, which weren't discovered until 1987. These bodies are 30 to 100 nanometers in diameter. It's a body of fluid enclosed in a lipid biolayer. A lipid biolayer is 2 layers of lipids. It's that simple. Exosomes are released by cells. They're thought to be important in coagulation, intracellular signaling, and waste management. And they can be used as biomarkers to deliver pharmaceuticals. Can be used as biomarkers or to deliver pharmaceuticals. I thought you might want to see what they look like. Scale here is, that length of that line is 150 nanometers, and these little bodies over here are thought to be exosomes in figure A. Figure B has tags. So these exosomes have on their surfaces molecules. And CD63 is the 63rd cluster of differentiation molecule. And TSG101 is a molecule associated with the tumor susceptibility gene 101. And these molecules are on the outside surfaces of the exosomes. And this is just a check to make sure that we're picking up the exosomes with these specific tags. And I'd like to thank the authors Gallo et al. This paper was published with this figure in it in 2002 in the journal PLOS One. Study recruitment design. We planned, and I think we've already, in fact, recruited 16 controls. We're going to recruit 15 individuals with scleroderma and interstitial lung disease, and 15 individuals with scleroderma but without interstitial lung disease. And the study is going to require taking measurements in blood samples twice, at the beginning and then 6 months later. So it's not a very onerous project to participate in, and we're not asking for hundreds or thousands of participants. The study genomic design, we're going to use RNA sequencing, also known as whole transcriptome shotgun sequencing, WTSS, which uses next generation sequencing to discover and quantify RNA in a biological sample. So this is going to be applied to detect microRNA in our tissue samples. And unlike conventional microarrays, no markers are pre-specified. So we can do gene discovery. And we're not using a chip with a predetermined allele on it at a known locus. We're also going to use nanostring encounter analysis. This is going to be applied to microRNA and tissue samples. And unlike conventional microarrays, it does not require amplification, and so can be done with microRNA from a single cell. Of course, there's a statistician involved, me, and you know what we're like. You know, one is never enough. So I'm asking for a couple of million of cells for each tissue sample. Yeah, we're like that. You know, we don't want to spend all the money just on one cell. You want to be a little bit more cautious than that. The question that I need to answer is whether or not this study is completing with the existing and ongoing genome-wide association studies in scleroderma. No, it's complementary. Well, how so? Dr. Mays and co-researchers in Canada and Europe are looking at nuclear DNA sequences of genes, while this study is looking at sequences of microRNA, which regulates genes. The study is closer to the biomedical action. The distance between gene and biomolecular activity is much further. Is this study competing with the GWAS studies for participants? Again, the answer is no. Recruitment regions are different, and individuals can participate in both studies. How so? This proof of concept study plans to recruit 30 participants in British Columbia, not including the controls. The GWAS studies have already recruited 4,867 patients in the European Union, the United States, and Canada. Are we competing with this study, with the GWAS studies, for financial resources? Absolutely not. The funding sources and scale of funding are completely different. As a reminder, support from the U.S. National Institutes of Health for Research at the University of Texas was reported to be in the neighborhood of $8 million US and counting. Funding sources for this study. We have received $140,000 Canadian in donations, and they're already held on account. We're in the process of application for a matching amount of $140,000 from Genome BC. So the total anticipated funding for this project in Canadian dollars is $280,000. That's a lot less than more than 8 million. So here's the progress report for Saturday, the 11th of June, 2016. Skin fibroblast samples from controls and scleroderma patients are in the freezer at minus 70 degrees Celsius. Standardization of extraction protocols for total RNA, and microRNA represents about 10% of our total RNA in our body, from tissues using replaceable controls, easily replaceable control samples, is wrapping up. And recruitment of scleroderma patients with and without interstitial lung disease is slated to begin in July next month. So on behalf of Drs. Dunn, Brody, Holt, Ryerson, Wilcox, and myself, thank you very much for your time today. And are there any questions? Please use the microphones.
Just a question. A lot of people with connective tissue disorders, such as scleroderma, are women. And there was, I don't know how you factored in that piece into your statistical studies. Also the fact of women that have had children. So the issue of what they call, I don't know the science of it, but the microchimes, I think they're called, that are related. Also a question of blood type, like individuals with RNA negative as opposed to positive. Any correlations?
So there haven't been any correlations, and there's no stratification done for being RNA positive or not. When a rheumatologist makes a diagnosis, sometimes it's in contrast to what the antigen test might reveal. So that's one particular aspect of it. With respect to the disease, it was alluded to by Dr. Winstone, 90% of the individuals with scleroderma are female. So it's overwhelmingly a woman's disease. What we're not doing right is recruiting enough men into the study. So unlike other trials that are done for common diseases, where women are underrepresented and they have to work hard to try and get that equal representation, we have to work very, very hard to try and get equal representation from men. And we have to work so hard that typically we, in studies, do not do that. So if we can get somebody into the study, we just tend to ignore their gender and just recruit them into the study. It is a very rare disease, and as you can see, when you're trying to recruit 5,000 people, it's going to be very difficult because that's 5,000 in Europe, the United States, and Canada for these large GWAS studies. Thank you.
This gentleman will talk forever if we let him. Anyways, Kevin, Dr. Keane, I'd like to give this to you, and thank you so much. It's all double Dutch to us, but now I know what a statistician actually does in the medical field, which is awesome. Thank you very much.